Good afternoon, everyone. My name is Katrina Chapman, and today is June 7th, 2023. Today we are going to be discussing sperm maturation, and sperm development. First, we're going to start off by discussing germinal epithelium. What is germinal epithelium? Here, I have a beautiful histological slide of germinal epithelium. We're going to discuss what each section is. Here you can see in the center there's the lumen, and you can see where there's a darker kind of circle that's going on, and that's going to be your myoid cells. The next section in front of the myoid cells, I want you to keep thinking of a circle. So the most inner part of that myoid cell line is going to be your spermatogonia, and the next is your primary spermatocyte. You have a spermatid, your spermatozoa, and then we have Sertoli cells, and we're going to talk about those. Your germinal epithelium for males is a layer of cells that covers sex organs in the developmental stages of the embryo. And keep in mind, when we're talking about germinal epithelium here in this discussion, it's going to be about male germinal epithelium because we are talking about sperm. As you can see, histologically speaking, the germinal epithelium has cuboidal cells. This is located in the testes. It covers the wall of the seminiferous tubules within the testes. It's located in these seminiferous tubules where spermatozoa are produced, as we can see. They're one of those layers within this circle. The germinal epithelium contains both Sertoli cells. Here's a Sertoli cell. And your developing spermatocytes. Sertoli cells extend from the basement membrane of germinal epithelium to the lumen of the tubule. These cells envelop the developing sperm cells. So again, here is your myoid line of cells that's circling. And then you have your different stages of the spermatogonia, spermatocytes, spermatids, and your Sertoli cells. And here in the center is the lumen. So as they're developing and moving on to their different phases of development, they are pushed more towards the center to the lumen, where they'll eventually be released. And we'll talk about that in a few slides. Sperm development. The seminiferous tubules are the site of sperm production. There are two types of cells. You have your spermatogonia and your Sertoli cells. The Sertoli cells are sustainment cells. They provide support while the sperm are developing. We also have the basal lamina. It's a barrier that keeps large molecules out but allows testosterone to enter. And then we have tight junctions. And part of the tight junctions, part of their purpose is to allow for blood-testes barrier, similar to the blood-brain barrier. And that, again, is to keep the other molecules and fluids out. We want to maintain the luminal fluid. The luminal fluid here is going to be high in potassium and steroid hormones, but it's going to have decreased glucose levels. Now we're going to talk about spermatogenesis. So here is a really great diagram from Silverthorne. And as you can see, spermatogenesis starts with signaling from the hypothalamus. So your hypothalamus is going to signal your gonadotropin releasing hormone here, and that's going to signal down to the anterior pituitary. Your anterior pituitary is then going to signal your follicle stimulating hormone and the luteinizing hormone. From there, on the one hand, here on the right, you see the luteinizing hormone is going to stimulate the interstitial cells, which then stimulate testosterone, and that's going to go to the body for the secondary sex organs or different effects. Your follicle stimulating hormone is really important when we're talking about spermatogenesis. It's needed to stimulate those Sertoli cells, which is where a lot of that development is going on. And then we have our cell products, the androgen binding proteins, and the steroid nutrients that are needed for the sperm to grow and develop and survive. And then we have the inhibin. So the follicle stimulating hormone is influenced by inhibin and activin. Inhibin and activin are different types of protein that are secreted, and they're going to help with that development of the sperm. The FSH is very important in spermatogenesis. Luteinizing hormone is also essential for spermatogenesis, but it's through Sertoli cell mediation. Here we have a diagram of a mature sperm from spermatogonium to division to sperm release. It takes 64 days. It's going to phase through the spermatogonia. Then it's going to go through meiotic division. It will divide twice. Then it's going to become a spermatid. The spermatids are going to develop flagellated tails. This will allow for motility once they're released. And then you have the acrosome, which you can see here at the tip of the head. And that has essential enzymes for fertilization, which makes sense because that head needs to be buried when it goes to fertilize. And then you have the mitochondria, which is here in the mid piece of the sperm body. And that mitochondria is going to provide the sperm with energy. And then at the end of those 64 days, your sperm is released from the Sertoli cells. Now, once released, the sperm are not capable of swimming yet. They have 12 more days of their journey where they have to make it to the epididymis before they're actually motile. And recall that slide earlier with the lumen and the different phases and stages all the way out to the myoid cells? That's staggered development. So as the sperm matures through development, that staggering allows for a continual daily rate of production of 200 million sperm. That's a lot. Now I want you to go ahead. I have attached a sperm pop quiz, as you can see. I want you to go ahead and label the sperm where the letters are, and also define the purpose of the labeled segment. And all of that you can find throughout these slides. And here are my references. Thank you for your time.